Hello everyone, welcome to our channel Medico Mentor. Today we're going to talk about TP53. It is a tumor suppressor gene. It is also known as guardian of genome, and why it is named so, we'll get our answer at the end of this video. There are different functions of TP53 such as it regulates cell cycle. TP53 causes the cell cycle to stop, and how it does so, we'll talk about it later. It helps in DNA repair. It also helps in cellular senescence or cell aging. And once the cell age, it does not divide further. And when DNA repair fails, it activates pro-apoptotic genes, and they give signal for cell to undergo apoptosis. TP53 is most mutated gene in the human cancer history. So virtually in every type of cancer, you can see TP53 mutations such as lung cancer, colon cancer, breast cancer and others. So the location of TP53 gene is short arm of chromosome number 17, region 1, band 3 and subband 1. TP53 gene encodes protein known as P53, and all the function that we listed earlier is done by this protein. So there are another family protein known as MDM2 family of protein, and the main function of this is to help in degradation of P53. So the activity of P53 protein is checked by MDM2 family of proteins. So in normal cell, what happens is MDM2 protein ubiquitinates P53. So what does it mean? Ubiquitination means adding ubiquitin protein to that structure, and if ubiquitin is added to that protein structure, proteasome is another protein which recognizes this ubiquitin tagged protein, and it helps in degradation of that protein. So basically MDM2 ubiquitinates P53 means MDM2 causes ubiquitin to be added to P53 protein, and once proteasome recognizes this ubiquitin tagged P53, it causes degradation of P53. So virtually in normal cell, P53 is undetectable. Whereas when there is DNA damage or there is cellular stress in the form of hypoxia, these are sensed by two proteins, ATM and ATR. ATM stands for ataxia telangiectasia mutated, and ATR stands for ataxia telangiectasia and RAD3 related protein. So these two proteins, when they sense DNA damage or hypoxia, this stimulates phosphorylation of P53 protein and MDM2 protein. So it means it adds phosphate group to these two proteins. When it adds those phosphate group to these proteins, that will disrupt the binding of MDM2 with P53. And what it does is, when MDM2 cannot bind to P53, it cannot ubiquitinate P53 protein, and proteasome cannot recognize P53, and thus P53 accumulates. Similarly, when there is oncogenic stress to the cell, RAS protein gets activated, and it further activates MAP kinase and PI3K AKT signaling pathway. These are the cell signaling pathway which, when stimulated, increases cell proliferation, thus creating cellular stress. And this cellular stress is detected by CDKN2A gene, and it causes increased expression of P14ARF protein. If you remember, this P14ARF protein is a cyclin dependent kinase inhibitor. Thus, it stops or regulates cell cycle. So what it does is, this protein P14A or alternate redeem frame protein, it binds to MDM2. When it binds to MDM2, P53 proteins get displaced from there, and P53 level accumulates. As we know, if MDM2 cannot ubiquitinate P53, P53 will not get destroyed, so it will, the P level of P53 accumulates here. So DNA damage, hypoxia and oncogenic stress helps in accumulation of P53, and when P53 accumulates, it will block the cell cycle. And how it does so is that it increases the transcription of a gene known as CDKN1A gene, and the function of this gene is to encode a protein known as P21 protein. This P21 is a cyclin dependent kinase inhibitor. So it will inhibit a formation of cyclin dependent kinase 4 and cyclin D complex. And what it will do is it will maintain retinoblastoma in active or hypophosphorylated state. Let me digress for a moment to explain you about retinoblastoma. So retinoblastoma is a protein which in active state is in hypophosphorylated form, which means less phosphorus is attached to retinoblastoma protein. So in its active state, it is bind to transcription factor known as E2F, and this cyclin dependent kinase 4 cyclin D complex. What they do is these complexes will phosphorylate retinoblastoma, which means retinoblastoma will be hyperphosphorylated, and this state is known as inactive state of retinoblastoma. In this state, now E2F no longer binds to retinoblastoma, it gets released, and once this E2F or transcription factor gets released, it will move to the DNA. It will transcribe those genes that are responsible for DNA replication as well as cell growth. And this cell DNA replication and cell growth are needed for transitioning from G1 to S phase in cell cycle. When P53 gets accumulated, then this P21 level gets increased. This P21 will inhibit CDK4 cyclin D complex, and if this complex is inhibited, then it no longer can phosphorylate retinoblastoma protein, and when for retinoblastoma is not phosphorylated, it will be in hypophosphorylated state or active state. It will continue to bind E2F, then E2F will not get released to transcribe those genes that will be responsible for cellular growth and DNA replication. Thus, that will block the cell cycle from G1 to S phase. Blocking of cell cycle provides breathing time for cell to repair its DNA. So the second function is DNA repair. When P53 gets accumulated, it induces the synthesis of proteins such as GAD45, which stands for growth arrest and DNA damage. This will enhance DNA repair. If there is successful repair of the DNA, then those signals that are responsible for P53 stabilization, that it ceases, then P53 level will return to normal and cell cycle will continue as in normal state. Third function is P53 induced senescence, which means P53 induced cellular senescence or cell aging. So when there is unopposed oncogenic signaling or unopposed hypoxia or shortened telomere. So let me tell you a little bit about telomeres. So these telomeres are attached at the ends of each chromosome, and cell has got limited amount of divisions before it ages. So every time the cell divides, the length of the telomeres gets shortened. So after a few divisions, the telomeres are no longer left to be shortened, then the cell ages. So these signals are sent by P53. The advantage of this cellular senescence is that though the cell are not normal, these senescent cells are not normal, but they further cannot divide, which means these senescent cells cannot develop into tumor. Fourth function is apoptosis. When the mechanisms described above are unable to repair the DNA, then P53 also helps in transcription of pro-apoptotic genes such as BAX and PUMA, and these genes will activate apoptosis, and there is cell death. So the advantage of this apoptosis is it will protect these damaged cells from transforming into neoplastic ones. So in conclusion, we can say when there is TP53 mutation, there is loss of P53 protein. But if somebody has got normal TP53, then it is also known as TP53 wild type. but he has got MD-M2 amplification, then also there is loss of p53 protein. And when there is loss of p53, then DNA damage goes unrepaired. Then it results in mutation accumulation, which increases the chances of malignant transformation. They can be inherited germline TP53 mutation and somatic TP53 mutation. So basically, TP53 mutation, TP53 gene, which is located in chromosome number 17. It has got two copies, and this two copies of the gene is known as allele. So, if somebody receives a defective or mutated TP53 from either of the parent, then he or she is said to be suffering from syndrome known as Li-Fraumeni syndrome. So, the person suffering from this syndrome are susceptible for broad spectrum of tumors such as sarcoma, breast carcinoma, leukemia, and brain tumors. So thank you. Thank you for listening. This concludes our today's video on TP53. Now we know how important is TP53 and why it is named guardian of the genome. If you like this video, please subscribe to our channel and share this video to your friends. Thank you.